lot of wells there. Okay, 10 and 10, total 20 wells. Okay, this solution was prepared. It was a liquid before. This is Agarose Gel, and so we prepared the Agarose solution, then put it here to make it solid. When we put that one, we put some comb here to make those wells. Okay. This buffer, this is a buffer. This is called running buffer, in which the gel will run. Okay. Here you can find some kind of wires here. These are electrodes. So we will connect, this red is positive, so we'll connect the red here. Here is the red point, and this is the black point. We'll connect this one. Then we'll start the electric current here, and probably around 110 volt we'll run this one. So this is called power pack or power supply, which will provide the electric current here. Now the DNA is negatively charged. If it is negatively charged, it will move to the positive side. Okay, so this is the positive side, and so our starting point is this side. So it will move to that side, to the positive side. So always the flow is from this side to that side. And this running buffer is connecting the whole thing, these two chambers. Here is a positive electrode, here is negative electrode, and this is the media for the electron transfer. Okay, this is the basic thing. Then I will go for everything in details, but you need to load the gel first. So here we have two samples. One is this is called standard marker or ladder, DNA ladder, and this is actually our sample to load. We'll load just 12 microliter per well. Okay, not more than that. How to do that? So at first, I'm loading the ladder here, and while loading, there will be no bubbles. So when you are using the pipette, press the first one, take the sample. Do not press twice here. Okay, so press first, take the sample in this way, go to the well very properly, and while you are going to the well, the main thing is that your hands should be steady. So put the hand here, and here is a support to load the gel. Otherwise, your hand will be like this while near the well. It will be like this. So hold it steadily. Then this is a very small well. So put the tip almost at the bottom of the well. Don't touch the bottom. And release with the two presses. Just release the whole sample. But after doing the second press, do not release there, because it will create bubble, or it will take up some sample with this. So just after pressing that one, just take it out, then release it. Okay, this is the way to load the gel. So I am doing the first ladder, then you will do the sample, and you can put another ladder also if you want. Just with the one press now. And just look at this thing, that it is not spreaded anywhere. It is just at the bottom of the well, because if the sample is going to the buffer anywhere, it will create bands anywhere in the gel. So it should be very, very cautious that the sample is in the well only. Okay. Now, from one by one. Come on. Go to the first. Okay.
So take this right here.
Only one place. Now go to the second.
So I got
I'm just imagining if someone got a muscle spasm right when they
I
want to be a doctor. You want to be a brain surgeon right there.
Here for microsurgeon.
I'll bet yeah.
You know, passes out doing this.
Yeah.
I passed out checking my own pulse.
Next, come on. Who's next?
That is. Wow.
My dad passes out to the side of the blood.
I know that I got lightheaded when the person took like one test of blood out of me.
Yeah. Well, I think that's
I
why.
donated blood when I came to class. I came into this class and I was like, because they like make me, they pick off so much blood. Yeah, and the tube's like huge. You can feel like how warm it is.
Yeah.
And it's
Oh,
like
really?
weird.
Oh,
I
wow.
remember when I had an IV once and it just felt ice cold.
Oh, well that's because they're pumping it in.
I know, but it felt like I could not get warm. I was like,
Yeah,
why
no.
is my that was
When I
like
got an idea, I like had asked for little blankets. I had like little blankets on me. These blankets. I was like,
I could get my blood taken, no problem, no problem, no problem. Donated blood, whatever, whatever. And then I got a concussion and I had a thing for like a month. And they gave me fluids and everything. Like, if you leave the IV in for like more than 5 minutes, I'm lightheaded and feel that fast.
there's the so thick.
Yeah,
It's like,
Oh, yeah, 'cause it's got a thick.
it's hollow.
Oh, I just remember.
It's a really big hollow needle. It's
I
weird
just-
that like they just stick that in your arm and you're not like.
I remember when they had to give me an IV doing this the first time.
Yeah,
My veins were all.
That's never
Digging it.
happened.
Like they put half of it in her arm and they're just like digging to try and find
Where
the
is that vein?
I
They
always
just
had
went
them-
to. They stuck me and then they were like, wait a minute, I gotta go to the.
you know, where all the heroin is, but I'm afraid of needles, so I'm not worried.
I do not look good. They give me a. I'm still questioning why I feel like, hey, it's a good idea to stab myself with a thing. I'm not even mad.
It's just like
I have
I'm just
no.
injecting randomly.
they. I don't know.
I have one
It's we have
they
proof that
always
it
get and like I
fucks
just
you up.
you can see.
Yeah.
So I have that one right on the surface.
Why do people keep doing it once they already know?
Yeah, like it's not like it's something new. Like
That used, actually, heroin used to be considered medicine back in the 80s. So that's,
I mean, there's a lot of opioids in there.
yeah. I don't know. It's time to act with the pill and this one. It's recording, so I can understand. Still can't see. I've seen
I did.
I don't even know. I'm gonna get in depth.
What's wrong with DC? Which is really cool. I was like, yeah. That's how I go. Like,
You don't
whenever
really-
I got the chance, I was like, yeah, I'll do DC.
But not
Yeah.
so food. The big decision.
Next.
What's his guts?
Spooky stuff. Thanks, class. I feel like I just need to leave.
I'm just really tired. I just kind of seems like I was like I just can't stand if I'm asleep for all and then like
I'm gonna be gone all weekend, so I can pack and stuff.
out with the light and then no, like actually like it's been so little about this one. Look at myself. Like what is that? Like why am I saying? Do I just not sleep at night? I don't know. I had a cold, so I've been taking Nyquil and I've been getting really sick.
Dang.
So what do you?
I need to check some height.
say crap. I felt like I
worked all the way up.
I felt like I had like
So like I have to do my own stuff.
I literally drank it and I was like,
After that,
like it immediately.
start working like postseason stuff.
Well, she said no time.
Yeah.
Yeah.
Yeah, that's so crazy.
That's what
I wonder what would happen.
I think you are working.
Anybody left?
I need to.
So last year, they did and then like it's, I think they made 905 or something like that. But I was talking to some management people this year. And they're like, oh yeah, since we had trouble keeping my parts out, we're like raising the pay. What's minimum now? Minimum is like 7 modi.
Here, that's 9 something.
Yeah. 905.
I thought it was 8 something.
But I know. Yeah, it was. I was told the way that the raising the pay to keep people. So it's something
You need to mark your length. Where is your something? Okay.
I can't.
So this is gel A, the first one. This is gel B. All the girls are in B group. All the boys are in A group. Okay. Now the length of the first lane is 1. So from here 1 to 10. Okay. Now you need to mark which lane you are. Do you have the idea of which lane you are?
Yeah.
Which lane?
Maybe I don't know where I am.
You can do 2.
You were,
Yeah.
Another one.
I don't know. I have to use.
Oh yeah.
yeah, but what should you go on the left or the right?
No.
Yeah. Well, as long as we
Well, I think you're B10.
we didn't
Were you on the end?
No, it's somewhere in the middle.
This is first one is only here, right? This is 10. So you are in 9.
I got raised.
You are in
I don't know.
from 19 to 1, 2, 3, 4, 5. You are in 5.
I'm in 5. Apparently.
What did he say?
You are 7.
I don't know.
No, he's confused because she's in the middle. I'm the one left there.
I was somewhere in the middle.
All I know is she was in the very middle. He told me to sit. I didn't know how to mute it.
I apparently am 5. That's all I know.
He told me to mute it. How is that? You're okay, so 7 and you're
How
8.
am I 5? You told me I'm not confused.
Just look.
Anybody wants to do? Anybody?
Go ahead.
Good. Are we doing this? One week? No, we can do it.
Yeah, they changed that company policy. It's a little dumb, but it's
I know,
like,
I
it
don't
wasn't
know
fair.
if you remember, but
But how am I supposed to know what I put?
the thing is, thought I think I got perfectly because it filled in all.
And
I did it. I didn't get all of it out.
Yeah, that's true.
I am.
Just like
I just struggle. They okay.
Inside the well a little bit.
A 40-minute drive.
like 20 minutes worth. No,
I'm like 2 minutes.
probably is. Oh yeah, it's probably.
Do you know where, how far?
How far is the
Okay, now I know where Coney Island is.
Yeah.
So what we are doing right now, we need to put this lid and and this will connect to the electrodes. Electrodes are already connected here.
Yeah, it'll take me 15. Legally, it's take me 15.
I've gotten the Amazon at 20.
Way I drive.
790 the whole way.
On it, turn on.
Legally, way I get 7.
Set the voltage to the root.
You can drive as fast as you want. You know where all the pop shops are. That's the thing. You just gotta memorize it.
Yeah, we'll use 110. That is fine.
Pray to God they don't have to release all the power up.
That is fine. Timer I am putting around 45 minutes. Okay, so it is set already. So our volt is 110. Ampere is set. Volt, ampere, and I'm putting the timer. Generally, sometimes we don't need to put the timer. We need to see how the gel is running. When I will turn on this one, right now I just set up this instrument, but I didn't run the gel. When I'll start running the gel, you will find from these electrodes, you will find the bubbles. If there is bubbles, that means it is connected. If not, then it is not connected. Look at it.
Now, Sam.
So after just 15-20 minutes, you can come here and see how the gel is going onto that side. Okay. In the meantime, we'll go through everything. Oh boy.
Yes.
A month off.
Huh?
Movie or something.
And your gloves should be here.
Which a shift is 4 hours. Now, like I said, reuse is 104. Saturday, we had 200. Almost double.
Up.
So they can do this thing called flex up.
Yep, we're getting.
So like really you only have to work 4 shifts and you will get your time.
4 shifts and look at your time.
Your 20 hours.
Oh yeah. Yeah, I mean that would be totally
And they they've got
What you need to do today, I will go for all those things, that how the gel was prepared and how to run the gel, what are the buffers, composition, all those things. And I will put it in the blackboard also. So we need to write in this way. So how the DNA was prepared, actually that is not our deal into this class, because to prepare the DNA, we need to cut it in restriction enzymes. Then we need to do the PCR to make multiple copies of the DNA, the amount. Then we can use that one. And this is plasmid DNA. Okay. But before that, I need to talk about this one because you need to submit the whole thing. Okay. For the lab reports, first thing is that read the instructions. Do not write that do it. If you this time write the do it or do this one, take the beaker, I will just deduct marks. I want to see what you did. Either say we took the beaker as you are working in the group, we took the beaker, we measured the things like that, or the best thing is that in a scientific writing, always write like that the beaker was taken. Who did it? I don't want to know. But I know that you did these things. Or it was measured with the spectrophotometer like this. So please check these things first. Second thing is that check all accessory documents. So we have like so many things here. The instruction, notebook instruction, the first page, your name, title, and everything, and your homework. If you have some kind of loose sheets, you need to put inside the uh, lab reports. So check all accessory documents. Next is check documents in each section. That is, if you have some kind of process and other things, put it there and make the cross reference to your writing. Okay, that suppose you cannot do the crosses, genetics crosses in computer, so you need to do it in hand. So put the crosses there and put the cross reference in your writing. Okay, the cross reference in this page, my that is, the cross is here and here is the writing. Okay. Check title, date. Date is the main thing. I didn't find the date in last couple of lab reports. So date is the main thing. And your group information and other things. Add tables and graphs wherever it is. Okay, because I didn't find a lot of graphs there. In your lab manual, we did those graphs in the class. In your lab manual, it was written that put a graph there, but in report there are no such graphs. Okay, so put those graphs there. And tables, graphs, pictures, figures, everything should have a caption. It should not come from the heaven or anywhere. Okay, it should have some caption. So this is this thing, and no unnecessary things there. If it is so, you cannot put your face figure there. Okay, I am this person, I did this thing. Not like that. So put something there which is relevant to your report. Put there, put a caption there, and mention that figure number or table number within your text. Now you are matured writer, so I don't want any kind of this kind of mistakes there. Now, pictures and figures must be levels. So if you have some kind of like chick cell, do not just put this picture there. Mention it. This is nucleus, this is cytoplasm, this is cell membrane. We need to know that. If you cannot do it in computer, you can do it in computer. Just put it in PowerPoint, the whole picture, put the arrows there, and you can write there. Then make it PDF or as a picture format, put it in Word. Or as a PowerPoint slide, you can put it in Word. It is possible. It might take a little bit more time. If you don't want to do that, just take this print, make the caption, take a picture in your phone, scan it in a PDF format, and put it there. Don't do JPG. Do PDF. That is a very good thing. The file size will be reduced. In graphs, you must have x-axis, y-axis levels. What is there on x-axis, what is there on y-axis, we need to know that. So when you are presenting through a graph, you must indicate what are there. And the legend. The bubbles or colors or the graphs lines, what are those things, you need to put a legend there. And it is generally automatically generated, that one. But don't miss those parts. And add all question answers from your lab notebook. We already, after each class, we went through all those questions and everything. So you need to put all those questions and answers in your notebook, okay? So, and must include the calculations. Particularly for, suppose like vitamin C, that is a good thing. Already you have the questions in your lab manual that what is the concentration of the vitamin C, what is the amount of vitamin C per 100 ml. So you need to include all those things. Then for microscope class, we did two things. One is the parts of the microscope. In your manual, you have a picture of the microscope and everything is there. What are the parts? You need to include that picture in your lab note. Okay, just take a picture of that one, scan it, put it there, and label it. Okay, different components of cells must be labeled. and you must identify different stages of mitosis. So when you did the mitosis, I gave you one caption there. These are the points, hence this is the stage profile. In exam, I need the same thing also. So if you are provided with the metaphase or anaphase cells, then you need to identify with the characters. So these things are there. That's why it is anaphase. This is the scientific writing. You cannot just say this is anaphase, this is telophase like that. So you have to show some characteristics that in metaphase all the chromosomes are aligned in equatorial plane. That is the characteristics of metaphase. Okay, then you can say that okay, it is metaphase. Like so, just put 3-4 points from your book. You can get that one, and what I gave you, there is the points also, that table. You can find the points from there. Only the visible points, which is there. You cannot see the spindle fiber, but we assume the spindle fiber is visible. So spindle fiber is always, should be always there. And for spectral lab and gel lab, add pictures of your thing, then add standard curves for each and everything and the results. For gel, you need to prepare the standard curve again, and for spectro, you already did the standard curve, so use the standard curve, then the unknown solution. Using that standard curve, that should be your result. So first, there are two standard curves. One is for your food dye and another is for the methylene blue. And those unknown solutions, based on those standard curves, you just need to put there. So if it is food dye standard curve, then the unknown food dye solutions, and if it is methylene blue, then the unknown methylene blue solutions. Okay, and for gel, we'll do in the next day that how to calculate the RF and everything. So for gel, one thing is must, that after the gel is done, you need to take the picture of the gel. Next day, just print that picture in a 6:8 format, not too much, just in this size, not the whole page or anything, and take a print of that picture within your normal page, normal white page, that you have some kind of space on both sides. So put the gel figure in the middle, and then take that paper with here, and you can take 2 copies. If you miss one, then you can use another one. Okay, because we need to write so many things just by the sides and everyone, and based on that we need to prepare the standard curve. We need to find out the molecular weight of unknown band. And in exam, if there is a such, you can have that kind of question, that I will provide you a picture of a gel, I will provide you the standard curve formula there. You just need to find out the band distance and to calculate the molecular weight of the band. Just these 2 words. I will provide the standard curve. You need not to do the standard curve for spectro and for gel also. Okay. And for genetics lab, use all crosses and everything. And in the first part of the genetics lab, there is corn genetics. That what is there, and human genetics, the autosomal traits, different kinds of traits. There is a table there. You need to add that table also. Okay. If those are not in proper order, there will be deduction of marks. So this time, last time I got a lot of problems for a lot of lab reports, because something is not there, whether the instruction is there, but the whole instruction is in the lab report, but not in the lab report. Lab report instructions should be in your notebook. The notebook instruction, report instruction should be on your report. So there are 2 instructions I provided in the first time. So one is for your notebook, how to write notebook. One is for how to write report. So the notebook part should be in your notebook. The report part should be in your report. There should be one contents page everywhere. The safety page, lot of safety page was missing in the last time. So safety page, the smaller part will be in your notebook, and the larger part will be in your report. And maintain all the documents properly, in proper order. Okay? Yes.
So you said after lab, you should answer those questions in the lab, right?
Yeah. Not notebook, report. Everything is in the report.
Okay, it's like after the conclusion
You can do after conclusion. You can do after conclusion, that's fine. But I suggest do just before conclusion if you can, because in sometimes when you are writing those things, uh, you might not have that much space to do that one. So you can do it before conclusion or after conclusion. That doesn't matter.
For vitamins lab,
Yeah.
you never use like the lemon juice daily.
I didn't give the lemon juice.
Yeah, you say you're gonna you're gonna
Okay, okay, then I will provide it. I'll let you know. I have this data, but I will need to. Any question? And next week it is due. Thursday. Because Tuesday you have the proof, which was extended. Right? Okay. Any more question about this? Okay. And I will provide you this one. This is the Electrophoresis Lab. So, couple of things. What is plasmid? We just need to have some basic idea. What is plasmid? Gel stuff. Sure. Any other idea?
Like a thick
Liquid.
liquid. Like a very thick, like
Thick liquid is plasmid. Okay. Any other idea?
Science.
Gel is plasmid. Okay. Any other idea? Yes. So you are reading something. They are just confused with plasma and plasmid. Plasma is the thing that is within between solid and liquid. The solid and liquid structure between solid and liquid is called plasma, which is gel structure. Okay, but in plasma TV is not like that. Plasma TV is something different. Okay, but this is called plasma. Structure between liquid, it is not liquid fully, it is not solid fully, like the gel structure, it is called plasma. And plasmid is a DNA, extranuclear DNA of bacteria. That is called plasmid. The structure of plasmid is circular, always, generally. Yeah, bacterial DNA is circular, and this is double-stranded DNA. It is outside the nucleus. And as it is outside the nucleus, it is very easy to isolate this plasmid DNA. That's why plasmid DNA is very, very important in case of biotechnology. So in case of E. coli, we got all the sequences of this plasmid DNA. We know where is the antibiotic site, where are those sites, we have all those things. So what we need to do, generally in biotechnology, suppose, do you have the biotechnology class yet? I don't think so. So in biotechnology, what we are doing that, suppose we need to prepare insulin. So we have the insulin gene in our body. So from human, if we can isolate that insulin gene from our DNA, and if we can put the DNA into plasmid and culture the plasmid, we can develop a lot of insulin. This is called biotechnology. So isolate the gene from human DNA, suppose this part, put it there inside the plasmid, because plasmid is much easier to access as it is extranuclear, it is on the cytoplasm, just isolate it. How can you isolate it? This was in your first class in the lecture. How can you isolate the DNA? How can you isolate the cell organelle? So, what is the process? Not here. You cannot find it. You need to read the Campbell's, okay? What is the process? This is called centrifugation. while doing centrifugation, you can have a different kind of speed. At different kind of speed, different particles are precipitated. From that precipitation, we can find out this. But before that one, it is not so easy. Before that one, we can use some kind of protease enzymes, some kind of other enzymes to digest out all the proteins and everything, so the only DNA would be there, and then we can centrifuge that DNA and we can have it from the either supernatant or from the other pellet. Okay? So centrifugation is the process to isolate the DNA. Now, this plasmid is called vector. Vector, why is it called vector? Because this is a vector for our insulin gene. Now it is in the bacteria or insulin gene. Now put that bacteria into the culture. Within 1 hour, there will be 1 million bacteria from one bacteria. Okay? So in a culture media is something like pinkish color generally, and having all the supplements, meats, which is required for the growth of the bacteria. So this is called culture media. So you can put it in the culture media, you can have some kind of like 1,000 millions of bacterial cells. They will secrete this insulin when during their protein synthesis. So are you having protein synthesis right now? I think so.
What?
Protein synthesis, transcription, translation.
We just finished that.
You just finished that? Okay. So during protein synthesis, it will synthesize this insulin, and so it will be in the culture media. So take out that culture media and then isolate the insulin from there. This is called biotechnology. We are using this plasmid DNA here, and we have two kind of things. One is human DNA is there, another is the plasmid DNA. But the thing is that we need to cut it in the specific area to isolate these two. To cut it, to cut a DNA, what kind of enzyme doing it? It is in your DNA application. There are two types of enzymes. One is called polymerase, another is called nuclease. So there are two kinds of enzymes you can find everywhere. One is called polymerase, RNA polymerase, DNA polymerase, DNA polymerase 1, 2, 3, DNA polymerase alpha, beta, gamma, epsilon, all those things in eukaryote and prokaryote, RNA polymerase 1, 2, 3, and some kind of exonuclease and endonuclease. DNA polymerase 1 has exonuclease activity and endonuclease activity, while the DNA polymerase 2 has only endonuclease activity, not the exonuclease activity, this kind of things. So nuclease is for cutting the DNA, and polymerase is for polymerization. What is polymerization? Adding the same molecules one after one. That is called polymerization. RNA polymerase, adding the RNA nucleotides. DNA polymerase, adding the DNA nucleotides. DNT. So these are the polymerase. And ligase is another enzyme for joining the nick junction. That is a very little part. That is called ligase. Here we have one enzyme called restriction endonuclease. Now what is the difference between endonuclease and exonuclease? Endonuclease is inside of the DNA. Exonuclease, that is endonuclease actually acts in between DNA. But exonuclease is just one part, side of the DNA, another side of it. So this is called restriction endonuclease. As it is endonuclease, that means it can cut any part of the DNA. Why is it called restriction? It can cut in a particular area. So suppose we have one restriction endonuclease which is called ECO R1 or Hindi3. They have specific sequence to cut that one. Suppose the sequence like A, T, C, C. It will cut in this area. And we have hundreds of restriction endonuclease. Each and every restriction endonuclease is specific for cutting a specific part of the DNA. using that restriction endonuclease, we can cut where we want to do that specific sequence. So, using that restriction nuclease, we can cut this part, the human DNA, from this plasmid, and we can isolate the plasmid DNA as well. Here we are using these kind of things. One is the human DNA, one is the plasmid DNA, and they have two different molecular weights. So we can get two bands here. Okay, one is for plasmid DNA, one is for nuclear DNA. Okay, this is the part of restriction endonuclease and plasmid. Now, how DNA molecules can be measured? So it is not molar, it is not molecular weight, but DNA can be measured in a very different way. What is that? Do you have any idea? Sequence is different. Sequence means what are the nucleotides in the DNA. But DNA fragments, suppose there are two DNA fragments. How can we measure the DNA fragments? Do not look at the front. This is called base pair. DNA can be measured in base pairs. So when you are, suppose later you will do the protein synthesis part, there you will find the tRNA. tRNA is suppose 80 base pairs long. So DNA, RNA, all those are in this way. So 10,000 base pairs long or 10 base pairs long, something like that. So what is base pair? What is base? What is base? Again, the question is that when you are talking about base, I need to know what is nucleotide, what is nucleoside.
Nucleotide is
At first start with base. What is base? Is that? That is not letters, those are the chemicals. Okay? And there are two types of nucleotides. One is called purine, another is called pyrimidine. So purines are double structures, double ring structures. Pyrimidines are single ring structures. Purines are adenine and guanine. Pyrimidines are cytosine and thymine. Okay? So cytosines are single ring structure, just like this. And purines are like this, like this structure. Okay? Now, the question is that in your DNA structure analysis, we have found that if these two type of chemicals will join together, it will be larger. If these two types of chemicals will join together, it will be small enough, but the whole DNA strand is parallel and will equal length. So the possibility is that it can bind with this one purine, one pyrimidine. And the band is A versus T and C to Z. There is no bonding between A to C, A to G or anything. This is the bonding. Okay. Now this is the basis. So nucleotide basis. Adenine, guanine, thymine, cytosine. And now the question is that what is the difference between nucleotide and nucleoside? Do you have the DNA structure class yet? So DNA is composed of these bases and a sugar, deoxyribose sugar. In case of RNA, it is ribose sugar. In case of DNA, it is deoxyribose sugar. The nucleotide base is same in the both cases, and both have a phosphate group. Okay? So the phosphate, sugar, and nucleotide, this is called the nucleotide, the base. Phosphate, sugar, and base. This is called the nucleotide. And nucleoside is just this part, without phosphate. So with phosphate, with ATP, DTP, CTP, they can freely move in the cytoplasm, in the triphosphate form. But when they are adding to the DNA, they need to be reduced to the monophosphate form, and then they can bind with the phosphodiester bond with the DNA. So this is the basic thing. Now in DNA there are double strands, two strands, okay, and those two strands are parallel, anti-parallel. Anti-parallel means if there is a A here in one strand, if here is A, here should be T. They can make the bonds. If here is C, it should be Z. D, A, like this. Okay? Now, if we need to measure the length of the DNA, either we can say that nucleotide long, so this DNA is 3 nucleotide long, right? Now we can say this DNA is 3 base pair long, 3 BP long. I can say that. Why?
Because you only have 3. Like,
We have 3 nucleotides. I can say 3 nucleotides long. Either we can say 3 base pair long. Here is the twist in the word. Why we are not saying 3 nucleotides rather than we are saying 3 base pair?
the spaces can be different.
This should be different. Should not be different. In case of RNA formation, they have the poly A tail. Poly A tail is 30 nucleotides long A. All are the A's. In case of RNA, we cannot say it is base pair. For DNA, we can say only it is base pair. This is 3 base pair long DNA. These two are paired bases. So if I say 1 nucleotide long, that means there is 1 base pair of A and T. If it is 2 nucleotide long, then it is base pair 1T, AT and CG. So there is always a pairing of bases in both strands. So that's why we can say this is a base pair long. So when we will measure this molecular weight, we can say that DNA is suppose 1000 base pair long. So the segment of DNA should be indicated in base pairs or BP. This is the short form of base pair. There is no molecular weight, no gram molecular weight, molarity, everything. It is just base pair. Okay. Now what is gel? Gel is a media where this is made up of something. For the DNA we use agarose gel, for protein we use polyacrylamide gel, and these are some types of substances through which the protein molecules or DNA molecules can pass. So if they can pass through it, that means it is a net-like structure. It has some pores inside it. So this is a porous structure, a sieve-like structure through which the molecules can pass. And this porosity, that is the amount, size of the pore, we can make it according to our will. Okay? So suppose we are using 0.7% gel here. If we use 1.2% gel, that means the porosity is much smaller. If the concentration increase, then the porosity will be reduced. So we will have small pores. So why do we need this kind of porosity? Because if the larger it is larger pore, then the larger molecule can pass. If it is a small pore, then the small molecule can pass. Okay, this is the basic thing. What is the DNA gel? Okay. Now the thing is that, so if this is true, now think about this, that DNA molecules can pass from the lane where we started to the negative positive poles. It will move through the gel. Now when it is moving through the gel, there is one thing, that if it is heavier, it cannot move too far, and if it is lighter, it can move too far. Suppose you are pushing the table, it will take much more energy to pull this or push this. But if I can move the chair, it is very easily I can push this. So with the same energy, smaller molecules can go far, larger molecules cannot go far. So in a gel, if we have different kinds of molecular structure, different BP of the DNA, then this is our starting point, the lane. We will have this kind of bands. So when we'll have this kind of bands, particularly you can find in the standard, that is the ladder, what we use, you can find 6-7 bands like that. Generally these bands, suppose like 1000 BP long, this band is something like 100 BP. So the smaller molecular, this is just 100 BP long DNA, that means it is small molecule, it can go further, but this is 1000 BP long DNA, so it cannot go further, it will be in here. Okay, this is the concept, that is the high molecular weight fragments can stay near your origin and low molecular weight fragments can go further. Now, what is the difference between the porosity of the gel? If it is happening in the 0.7% gel, if I will use 1.4% gel, that means concentration is higher, porosity is less. Then we could find this band is very near because it cannot pass. And these bands are more widely separated. So that's why we need different porosity of gels. That if we want to separate the smaller molecular weight molecules, then we need to use high porosity gels. That is pore is very small, so we can separate the smaller fragments. If we need to separate the higher fragments, that is the heavier fragments, then we need to use 0.5%, 0.1% gel, so we can separate the high molecular weight fragments. Now, what are the things we are using here? So you can write just DNA sample provided, because this DNA sample was prepared in some other way. This is a plasmid DNA, so that's why we cut it with the restriction enzymes. Then we did the PCR to make it multiple copies. Then it was prepared in a solution. So it is a different thing. So you can just write DNA samples provided. Okay? And we use the ladder, standard DNA markers. And this standard DNA can be purchased from any company, DNA companies, biotechnology companies. And these are not really that much expensive. So you can use, suppose 10,000 to 2,000, whatever you want, that kind of ladder you can purchase from these companies. And generally they would say that what kind of agarose gel you need to use. Okay? Because that the porosity differs in the separation of the gels, different kind of bands. We have 0.7% agarose gel here. That is our electrophoresis unit where we are doing the electrophoresis. This black one is called the viewing unit. It has one kind of UV light inside it. As we are using the fluorescent brand Ethereum bromide there, we'll take the gel, we'll put there with the UV light, and UV light is not good for your eye, so we need to cover it with the black thing. Then we can view the gel, the bands there. It will be fluorescent band. We have two kind of buffer. One is called a running buffer and one is called loading buffer. Okay, running buffer is the huge amount of buffer what we put to submerge the gel. That is the running buffer. And the loading buffer is the buffer what we used with the dye, running dye, that is called the running dye, with the sample. Generally, loading buffer contains a little bit of glycerol. That's why it is heavier than the running buffer. Okay, why do we need to make it heavy than running buffer? Why? Yeah, so it can stay at the bottom of the well. It cannot go overflow with the running buffer. That's why we need to make it heavier so it can stay in the bottom of the well. Because if you put the samples on the running buffer, it will make different kind of scatter bands here and there, everywhere. Or you cannot find any bands there. That's why we need to make these two kinds of buffer. And this is our power unit. It is saying that 1 hour, but we are using 45 minutes today, and I think 45 minutes is quite enough for that. And those are our safety equipments. We must need the gloves. Now, generally this buffer is called trace acetate buffer or trace borate buffer. These two kinds of buffer we are using. Here we are using the trace acetate buffer. These are the common running buffer for the gel. And this is the composition, trace base, acetate and EDTA. And make always, while making the buffer, the last lines will be add distilled water up to 1 ml. 1 liter or 100 milliliter, something like that. So the preparation of buffer is like that. At first, mix all those things, prepare a small amount of sample into a measuring cylinder, then add water up to 1 liter. So it is not that make the solution first and put it in 1 liter. The total volume should be 1 liter. And in future, if you work in a lab or anywhere, just remember these things. The buffer means up to 1 liter, not in 1 liter solution. Because when a lot of students are coming into our lab, they are making these mistakes. So they are preparing the whole thing there, if it is solid also. So they just measure the solid thing, solid EDTA, and put it in 1 liter solution, not like that. At first, measure those whole solid things, put it in a measuring cylinder, add a little bit of buffer there, little bit of water there, make the solution dissolved into that thing, then make it up to 1 liter. Okay. And I put two of them, that is a trace acetate and trace borate buffer. What are the compositions there? But we are using trace acetate here. And then preparation of agarose gel. So I'll show you a video how to prepare this agarose gel. I think we'll not go through this one. Just the thing is that we need to measure the agarose. Agarose is a solid powder. We need to measure the agarose in a, that is, what is that called? The measuring unit, balance. And that balance is very, very, not those kind of things. We need a very specific balance where we can get 1 mg recording. We need to measure the mass of those agarose thing and then put it in a water. Then we need to heat it up because it needs some heat to be dissolved, the agarose. After that, we need to make it cooler and we can prepare into the gel. I'll show you one video there. And for this molecular weight ladder, it is generally prepared. So when we will purchase those things, they are already in buffer and with a dye. And this is called running dye. So what is the use of this running dye? That we can see how the gels are moving, how the molecules are moving. And you did this thing, loading samples into gel, remove the comb, and after that, the best practice is that at first run the running buffer, whole. Dissolve the whole gel into the running buffer. It will take out all the bubbles from the wells. Sometimes to get a very good result, generally we run the blank gel for 5-10 minutes. So the pores will be very good, everything will be cleared, then we switch it off and then load the samples and then can run. Sometimes we need to do that. But you can do it in other way, that is the when it is a solid gel at that point, you need not to add the running buffer, you can load the sample first, then you can add the running buffer. But there are two problems there. One thing is that you can have a lot of air bubbles inside the well, or you can split up the samples here and there while putting the running buffer on top. So when the running buffer will come with this, here the samples are loaded. When the running buffer will come here, it will split a little bit. So that's why at first put the running buffer, fill the all well with the running buffer and then load the samples, and the sample contains the glycerol, so it will be on at the bottom. Okay, now Next part, we'll view the gel bands here. Take the picture of those bands. Next day, we'll prepare the standard curve, and from there, we need to calculate the uh, relative distance of each and every band and the molecular weights. Now you can check how the gel is running. After that, we go to the let's see the bands here. This is already output.
Just like I'm both looking for it. Sorry.
I don't understand what I'm looking for.
You see it's
The
moved.
rings?
It went from the wells to
Oh,
over there.
okay. Yes. It has moved.
Halfway.
This is the purpose of having this die here, this running die. Because based on that one, we can sometimes, we need to overrun the gel. We can take out some of the molecules to make the separation much better. Sometimes, in case of proteins, when we are doing the Western blot, we can just overrun the gel to get a particular band in a particular radius. Or if the two bands are very close in a normal way, we need to overrun the gel to make it separate. So for these bands, these kind of dyes are very, very essential to look at that how much the gel is there. Now we can see one thing here. Oh, it's off. Okay, that drops.
I just keep on recording this whole thing.
It turned itself off.
Is it done?
What did you say?
No more. Thanks.
I noticed everyone was looking at it.
Just like now, I'd be like,
Another thing. Look at this. Can you see the bands?
Are you people? Okay, you're not people. Gotcha.
yeah, so here it's like another
The purple is the light.
I look clean, but yeah.
Okay, I thought he said I was confused for a second. It looked like you were painting.
Cool.
Well, I still don't know what I was looking at, but I looked at it.
What you found?
I stared at something.
There you go.
What does it mean?
You know, means that we're almost done with class. Did you know what you were looking for exactly?
I know it was in microwave.
Did you find it?
Yeah, it was like the little white glowing yellow glowing band.
I don't even know. I saw some greenish color stuff, and I was like, I saw some little dudes, and I was like, okay. Wait, what? We're doing something? Oh,
So this is the structure of your gel chamber. What are the things
Fancy.
and how can you measure the bands? In which way the bands will go?
show a nice picture. Ooh, video. Go YouTube.
One of these very clean. I cannot make it.
Turn it up a little bit, like the sound.
Jeez.
Add that 150
Oh,
milliliters to a microwave safe container. Then we measure out the appropriate mass.
So at first take the buffer, put in a microwave safe container, measure the agarose, put it there.
We add the agarose to the TBE.
Agarose.
Dissolve it to its heating. So we close the container with a vented cap because we'll be boiling.
So we need to use a vented cap because there will be some bubbles.
Next, notice the labeling on the bottle, and then listen as the researchers boil their solution.
And here is the microwave where we need to prepare those things. Okay.
And the first check, notice the agarose is not starting to melt.
It looks like weird lemonade.
Stages.
Yeah.
Actually, the person who recorded, he has the microphone with him.
Uh. Looks good, guys.
Next, the solution must cool. Use a 55 degrees C water bath to let it cool. After
Just we need to make it at room
DNA
temperature.
dye
That's it.
Cyberscape comes as 10,000 times what we need to fix it. So we use about 1
Oh, you don't got none there, folks?
Here we are adding ethidium bromide in the gel, but here what we did, we added it in the running buffer. Okay, rather than using the gel. This is our total agarose solution with ethidium bromide and the agarose.
If it's too hot, go back in the water bath to
She has no. She's unsafe at the moment.
It can cause damage to our equipment. Here are a couple of examples. These are no longer suitable for use. When our agarose gel is cool at an appropriate temperature,
Then I have that kind of things that can be rotate into this chamber, so it will be tight. So when we need to prepare the gel, we need to do like that. This buffer we can add later also, there is no problem after preparing the gel.
insert the gel former into the box.
But see this one.
We recommend students use gloves when doing this procedure. But here you see
What
the
about
glass
you?
and some moisture just a bit to ensure that they seal well in the gel former.
Get your gloves. Get your
Notice
lab
how
coat.
the gel former is put into the box in this direction. We'll change it later.
Change it.
We're going to check to be sure that it's level. We need a level gel. Put the bubble inside the circles. We're ready to
That's an interesting level.
gel.
Now the question is that why we need to check the level?
The gaskets are sealed so our agarose doesn't leak into our
Because otherwise one part will be thinner, another part will be thicker. When it is thicker, then it will be difficult to move the molecule.
We call these cones. Then you see 2 cone formers are inserted into the gel former box.
Okay.
And then we'll be ready to put our agarose gel.
the volumes, you can have it
So
in
you're
your
going
home.
into the gel form.
Just use them.
When we're all done, you want to be sure to rinse out that bottle really well. First with tap water to make sure that none of the Agarose sticks in our bottle, and then always rinse our scientific equipment with deionized water to make sure there are no interfering pollutants in our bottle. It's always a good idea to keep that really clean, and if you're the last one to use something, of course you clean it and make it ready for the next person.
For gel, we are always using deionized water because there should not be any kind of ion, and DNS-free water
Gel box at the bottom
for
of
the
your
DNS.
or to the left as we turn, is solidified and is ready to be loaded. But before we can load the gel, we have to make a few changes. First of all, we're going to remove the cones and expose the wells. These are the wells into which we will load our DNA samples. So notice that the well is literally an indentation in the agarose. You will be assigned certain wells for your samples. Next, we have to turn this gel around so that the current applied to the gel box will pass across the gel. Remember, it's the movement of the current that facilitates the movement of your fragments of DNA. We put the wells to the top of the gel, in this case, towards the black electrode. It takes a few minutes to get the gel in place, and then the gel must be covered with a buffer. It's the buffer that facilitates the movement of the electron
They are
current
using
across
Tris
the
Boret
gel.
buffer.
Again,
We use
the
Tris
electron
acetate
current
buffer.
facilitates the movement of your DNA fragments through the mesh of the agarose. Next, listen as our researcher describes how she loads her sample into the wells.
Hand on the gel box and on my other elbow, so I'm kind of, and then you have to get the right angle to where you can see. I guess we're skipping the outer lane and going one in.
Now watch as samples are loaded into the wells. The tip of the pipette is held just at the very opening at the top of the well. If you are too far out of the well, the sample may bleed out of the well. If you are too far into the well, when you expel your sample from the pipette, it may come squirting out. The sample is stained with what we call a gel loading dye. The gel loading dye usually has a nice heavy molecule to make sure that your sample will sink into the well. Notice how slowly and carefully this researcher moves from loading one sample, changing the tip, and loading the next sample.
That is the kind of bleeding. If there is that kind of bleeding, the band could be different.
More samples being loaded, and then we'll show you the completed gel. When the samples are all loaded, we apply a current of 110 volts across the gel. Notice the display oscillates on our power source, but it's still working, and our restricted fragments are beginning to migrate across the gel.
Cool. Next video.
You want to watch this one, you can watch this one.
In this week's lab, we'll be doing DNA Agarose Gel Electrophoresis. This is a technique that is used to separate different sized fragments of DNA. DNA has a negative charge, and so all DNA will flow towards the positive electron. And fragments will be separated based on their size.
This one's a 17-minute video.
Faster through the gel matrix, and larger fragments will move slower through the gel matrix. I'm going to show you how we're going to set up our gel box. We're going to be using a mini gel electrophoresis box. There are several items here. We have a micropipette for loading our samples on the gel, tips for the micropipette, a tip waste bucket. We have our testing bowl.
Wait, you mean I actually got to teach?
Samples.
What are we doing? So, weird. It sounds very interesting. Oh, It's just all okay.
You can take the picture. You can take the picture of that one. Just take some more pictures.